Scientists have several ways that they use to determine whether organisms are the same species or different species. One of the most standard and oldest ways that we've used to identify species is to look at morphological traits. So the very first people to really start trying to decide if an organism was the same or different species used morphological traits. So these are physical characteristics: color, size, shape, all those kinds of different things. And so this can be useful because, as we said, the organisms that are the same species oftentimes do look similar. But it can also have some challenges, like, okay, which traits are important, right? So you can have things that, like, is color important for a certain species? Is the number of toes important? Is the shape of the ear important? So there's all these different traits that organisms have, and some of them are important for the species, some of them are not, some of them might just be variation within the same species. You also have to deal with traits that are continuous. So continuous means not distinct, right? So this was a term that came up with genetics, Mendelian genetics as well. But if you think about, say, black versus white coloration, like so black fur versus white fur, if you just have black and white, that would be two distinct variants of the trait. But what if you have gray and dark gray and light gray? Well, then you're getting into continuous variation where you have individuals at either extreme and individuals that vary across the gradient. And so that would be very hard to use to determine speciation. How do you quantify differences? Like, is how similar does the color have to be, right? Like, is it 100% the same? Is it 90% the same? Is it 85% the same? That's very subjective. That can be hard to put into hard numbers that everyone can kind of use the same system for. And then the other difference that you can get is that depending on the species, you can have a lot of variation within that species, and that's what this is an example of. So these are two frogs of the same species, but they're dramatically different colors. And these two frogs, on the other hand, are the opposite. They're actually different species, but they have really similar coloration. You also can run into things like sexual dimorphism, so males and females looking very different, yet still being the same species when it comes to their appearance, looking very similar. Or you can, as I mentioned with the eagle example, you can have juvenile versus adult phases look very different. So there's a lot of challenges to using this strategy. Again, not to say that we don't use it a lot for determining species, but there are some challenges. So in our toolkit for determining speciation, we have a lot of other things we can use as well. So one of them is molecular features. So this can be DNA sequences, it can be chromosomes, how the chromosomes are structured, chromosome number. It can even get into protein sequences or biochemical pathways as well. These are less common ways to do it, but we can also look at those. And so again, this is looking for similarities and differences among the populations. And this can oftentimes be used in addition to, like, for example, the physical appearance. So one of the advantages to using especially DNA sequence is that you can actually make a number. So for DNA, we can make 95% homologous. That would mean that the DNA was 95% the same. Or you can say, 50% homologous. And so this comes back to the idea that the more different they are, the farther apart they are in evolution. And so that might suggest that they're a different species. However, it is difficult to set a bar. So one of the hard things about this one is we don't have like a hard and fast rule, like it has to be 75% different in order to be considered a different species. For some organisms that we consider the same species, they actually have a lot of differences genetically among individuals. And then for other species, they have really small differences, and we still consider them different species. So for example, humans and chimpanzees only have like 3% difference between them. That's a really small percentage difference compared to other species separations, but obviously it has significant consequences. So there's not like a hard, fast rule that says 3% difference will be a huge difference. It just depends on the organisms involved, whether or not that will be enough of a difference to make it be a different species. Another strategy that's often employed is relating the species to its ecosystem. So every species has an ecological niche, like a place where it lives and the resources that it uses or provides within that ecosystem. And so organisms that are in other ways very similar might have different ecological niches. This can oftentimes too get tied to geography. So for example, you might have an eastern species of a bird and a western species of a bird. And they might look very similar, have really similar behaviors, but one is found in one region and another is found in a different region. This is one of the things that we rely on a lot with bacteria as well. So bacteria don't have a lot of DNA, so it's hard to use DNA as a difference. They don't have a ton of distinguishing physical characteristics. There's a couple that we'll talk about later on, but mainly they don't have a lot of traits, so there's not a lot that we can see. And so one of the focuses on bacteria has been trying to group them based on some of their ecological factors, like what food do they eat or need, what conditions will they grow in. And so that's been one way to try and group them and distinguish the species. Again, it's not foolproof. There's been challenges to that as well, because a lot of bacteria will tolerate lots of different conditions. So you can't classify them one way if they'll also live in a different condition. So again, it has its uses, but there's still weaknesses to this strategy as well. All of the ways that we classify species tell us about their evolutionary relationships, but in some cases we rely almost exclusively on their evolutionary relationships. So we make an evolutionary tree and we use that tree to determine whether something's a different species. And this is going to be especially common when we're trying to fill in species in the fossil record. So we're saying, okay, I don't know much about this species. I maybe have this many bones from it, but I think it's a separate species because it's obviously not the same as the horse today because it's extinct, but it must be a different species back here because it's in the fossil record that way. And sometimes DNA can be used this way too, like we look at the DNA and we look at a particular gene and say, okay, well, there's an evolutionary tree that shows this organism being a separate species than another organism. And so in a lot of ways, this strategy combines pieces from the other strategy. A little bit of the DNA, physical appearance, different things like that might get pulled into to create an evolutionary tree that shows speciation. And then the last one here, the ability to interbreed. So in many cases, especially with sexually reproducing organisms, or well, actually we can only use it with sexually reproducing organisms, we determine speciation based on the ability to successfully interbreed. Okay, so I'm going to highlight here successfully and say that the offspring, to be successful, a reproductive event must produce viable, fertile offspring. Okay, so sometimes we can have interbreeding, but if the offspring aren't healthy, viable, and fertile, then we would not consider that breeding within the same species. So the kind of thought process behind using this criteria for speciation is that as organisms separate over time and change over time, you get reproductive isolation. So again, this is the outcome of evolution. And all the different changes that have evolved will cause two different organisms to not be able to interbreed with each other anymore, and we would therefore consider them different species. This one, again, like there's some strengths to it, but and the strength really is that interbreeding can be a readout for lots of other evolutionary traits. So again, it's a readout of how similar the DNA is. It's a readout of how similar the physical traits are. But it has some challenges. It's hard to see in nature. We don't know who breed with who. We don't know what animals are always doing in nature. In some cases, organisms are capable of interbreeding, but yet they choose not to in nature. And so we sometimes know this because of animals in captivity or who have been manipulated by humans. As I mentioned, we can't use this at all for species that are asexual. And of course, we have no idea for extinct species as well. So those are the limitations of this strategy. So again, just to kind of wrap up this topic a little bit, when we're talking about interbreeding, we're saying that if they can't interbreed, they're reproductively isolated. And it's a result of evolution. And once they're reproductively isolated, there's a whole bunch of mechanisms that keep them isolated. So it's what's sometimes called a virtuous cycle. So they are isolated, and then they have these mechanisms that keep them isolated. So keep them from interbreeding from each other. Okay. So again, reproductive isolation is a result of evolution and a result of natural selection, all that other stuff. And then we have mechanisms that maintain that reproductive isolation. So an example of this is the finches. So we've talked about the finches and their beak size before. So due to natural selection, the finches have different beak sizes and shapes. And this was in order to feed, right? So this was related to fitness for feeding. But what they found is that as beaks changed, so as beak size changed, due to natural selection, it also changed the bird's song. So adaptation for food had a side effect of a change in song. Because birds use song as part of their mating behavior. Okay, so this is part of mate attraction. This changed who they would mate with. So different beak sizes contribute to reproductive isolation. So again, it's showing how reproductive isolation is caused by evolution and then is maintained over time because of these like mating behaviors, for example, in this case.